you are ready to charge forward with the rest of the semester. A few announcements, as always. One is that a reminder that Brain Explorers is the one extra credit opportunity in this course. If you're interested in staffing a station, there will be another one of the organizational meetings on Wednesday at noon, at 1 o'clock here in this room. So please join us to start to put together what are you interested in presenting to the public. So if you're interested in science communication, working, being part of a group that is actually national. Brain Awareness Week has already happened, but this is our contribution to Brain Awareness Week. And the Joseph Moore Museum is hosting it. It will attract kids and their parents from the community. It can be a lot of fun, so please feel welcome and invited to come help us organize this. The actual event is on Saturday, March 30th, but an organizational meeting is on Wednesday at 1 o'clock. Mallory, is there anything else I need to say? Feel free to ask me or Mallory or Morgan Bailey or Abe Shodhari if you have any questions. Let's see, another announcement is that there is a conservation biology symposium actually on the same day as Brain Explorers. So if you don't want to do Brain Explorers, there is a symposium going on conservation and field biology. I told Wendy I would promote this, so that's what I'm doing. If you'll see these flyers up around campus, and so if you're interested in conservation biology, here's something else going on campus on Saturday, March 30th. And one more time to remind you that if you are interested in fulfilling your wellness credit, but doing something valuable in biology at the same time, there is a wellness course that you can take in species control, getting rid of invasive species, working in the woods with Brent Smith. It is a wellness because it's hard work, but it is also fun to feel like you're helping out with getting rid of invasives. And so that's an opportunity for those of you who need to take care of a wellness. I think the registration for this has passed, and you might be concerned about a penalty for a late registration, but in this case, the registrar's office has waived penalties. So if you're interested in doing this, you can still do this with no penalty. And finally, a reminder that quiz 3 in this course is due on Sunday. It will be available here at the CST desk on Thursday, and it's due on Sunday at 11:30 p.m. as always. There's a thing in Moodle about how to focus your studying efforts for that quiz. So read that posting on Moodle. So what we're doing today is making a transition from sensory systems to motor systems. So, at a simple at the simple oversimplified level, we are input-output devices. We receive input in various sensory forms: visual, auditory, tactile, sensory, smell, taste inputs. We process those inputs, and then we execute outputs. And the most important output, well, I can't say the most important, one of the important outputs we have is movement in response to a stimulus. Oh, that smells good, let me go over that way. Oh, that smells bad, let me get away from that. A motor response to a stimulus. Oh, that sounds interesting, let me go over that way. Oh, that sounds like something I want to avoid, let me move away from that, that sort of thing. So we're gonna focus on the motor system. And here's a cartoon of the motor system, which is kind of fuzzy and hard to see, but the main points are that it's muscles, of course, that are the that do the operations of the motor system. Those are our motors, our muscles. And so we'll be starting here where we, learn about muscle action from cell physiology, a reminder of how muscles work, how they contract, and then start to work upstream. So we'll move from muscle physiology to neuromuscular physiology, how the nervous system activates muscles, and then how the spinal cord controls the motor neurons. It turns out that a lot of your motor system, a lot of the programs in your motor system are not in your head, they're in your spinal cord. So the spinal cord, the central nervous system of the spinal cord, the gray matter there, is going to be an important part of the motor system. Not just carrying the information, but actually processing the information and creating some of the circuits that are necessary for our motor activity. So spinal, action, motor neurons, muscle, and then we'll talk about the higher centers of the brain, the motor cortex, the cerebellum, basal nuclei, like the caudate nucleus, or the putamen, or the globus pallidus, or the substantia nigra. Centers in the brain, as well as cortical control of the movement. So we're going to start at the muscle and move our way upstream. Okay, muscle. There are 3 types of muscle, and we're going to be focusing on the skeletal muscle. So this is muscle that we'll be focusing on in this course. There is heart muscle, which those of you who've taken the other A&P class know about, and those of you who are yet to take it will learn about. But cardiac muscle is different from skeletal muscle. And smooth muscle is different still. Smooth muscle are the muscles that control the diameter of various tubes in your body, like blood vessels. Smooth muscle controls blood vessel diameter in the cardiovascular system, or it controls the motility of your stomach and your intestines in the digestive tract. So smooth muscle and cardiac muscle become important in A&P 2. We're going to focus on skeletal muscle. Skeletal muscle is, you've seen it in the lab. Skeletal muscle are muscles that connect to the skeleton. That's where it gets its name from. So you know that the biceps brachii is connected to the scapula. The long head is connected to remember. Anyway, the 2 heads of the biceps are connected to the scapula. One head is connected to the coracoid process, and the other head is connected to the top of the glenoid tubercle. That one I know. And then the other end of the biceps brachii is connected to the radius, actually. So when we move our biceps, we're moving bone. And so a muscle like the biceps or the brachialis is a collection of muscle fibers. So the cell type that we're talking about here are muscle fibers. Muscle fibers are the cell. Muscle fibers are large polynucleated cells. That is to say, they're long cells. Some of them can be inches long, maybe only microns in diameter, but inches long. They are formed by the fusion of embryonic stem cells that are called myoblasts. So the fusion of these cells that are sort of normal-ish cells in the sense that they have one nucleus, but the fusion of this leads to the development of these fibers that have many nuclei. It's still one cell, but it has many nuclei. Within the So the muscle fiber is the name of the cell type. Within the muscle fiber, so here's a blow-up of a muscle fiber. Here's a cartoon of a single, one of the many hundreds of nuclei in the muscle fiber. And then within the muscle fiber are myofibrils. That's the cytoplasmic structure that does the contraction. Myofibrils are made up of serial stacks of sarcomeres. That's a word that you hopefully, that rings a bell from cell phys. We'll talk about the different parts, the bands and the lines and the zones and the discs of the sarcomere in a few minutes. So going backwards, sarcomeres make up myofibrils. Myofibrils, bundles of myofibrils make up muscle fibers, and bundles of muscle fibers make muscles. Okay, more muscle anatomy. So here's the bone that the muscle is attached to. The tendon is what connects muscle to bone. The outside of a muscle is called the epimysium. It's that, I think on the cadaver you've seen the sort of shiny, whitish surface of a muscle. That's the epimysium. Sometimes the epimysium is covered with a sheath that is different, but the shiny surface of the muscle itself is the epimysium. Within the muscle, there are fascicles of muscle fibers. So a fascicle is a bundle of muscle fibers that is surrounded by an endomysium. So epi, out, endo, in. The names are a little helpful. And the bundles of fascicles, each fascicle is surrounded by a perimysium. So the perimysium surrounds the fascicle, and a bundle. So this is perimysium, and each muscle fiber is surrounded by the endomysium. Just anatomical names within the muscle. We have multiple types of muscle that I just want to get out there and talk about. We won't zoom in on this too much. So we have slow and fast twitch muscles. We'll get into what a twitch is by the end of this class. Slow and fast. And then the fast is broken down into two subtypes: fatigue resistant and fatigable. Okay, so slow and fast, fatigue resistant fast, and fatigable fast. Okay, so let's break this down. So the slow twitch muscles, they are slow to contract. So they have a slow contraction mechanism because they have a slow variant of the myosin that you'll remember from cell phys. The myosin is what's going to end up doing the work, the ATP-dependent work of contraction. Remember the word myosin and then actin. We're getting to those things from cell phys, but the slow twitch muscles have a slow myosin. That is to say, they're, the enzymology of that type of myosin is relatively slow. They have lots of mitochondria, so they make almost all their ATP by oxidative phosphorylation. Remember that from cell phys. And these are the postural muscles. So these are muscles that we use that, kind of all the time during the day. Muscles of the back that are important for standing upright. muscles of the leg that are used to stand upright. So right now, even though you're sitting, you're using postural muscles to maintain your position. And you're not exercising, but those muscles are working, and you're not even aware of it because they're working sort of in the background. So, and they're red in color. And so those of you who know chicken, right? There's red muscle in chicken and there's white muscle in chicken. The red muscle in chicken are the legs, because chickens stand, when they're standing around, they're using their postural muscles. Okay, that's the slow twitch postural muscles. Fatigue resistant, so fast twitch muscles that are fatigue resistant have a faster variant of the myosin. They have mitochondria, so they produce ATP by oxidative phosphorylation, but they also have a fairly large abundance of glycolytic enzymes, so they can produce ATP in the absence of oxygen if necessary. And so they're faster, but the fact is, by the production of ATP by oxidative phosphorylation and glycolysis gives them the ability to resist fatigue. Okay, so let me leave that just hanging there, because now let me talk about fatigable. Fast twitch muscles that are fatigable have the fastest form of myosin, and they make their ATP almost exclusively by glycolysis, which is faster than oxidative phosphorylation. And so they make all the ATP they need by the glycolytic enzymes. They don't have very many mitochondria. And so they can make their ATP fast, but because glycolysis is not very efficient, they run out of ATP fast. So if they run out of ATP, they fatigue. Running out of ATP is what causes us to fatigue, our muscles to fatigue. They can't work without ATP. So for example, in our body, the best example of the fast twitch, fatigable muscles are the muscles of the eyes. Our eyes move really fast. The eyelids, the lateral, medial, superior and inferior rectus muscles of the eye, we can move our eyes really fast, but it's not like they're lifting weight. So they don't need to be fatigue resistant. They are fatigable. Very much white muscle, fast twitch muscle. Fatigue resistant would be, the muscles of, a sprinter's legs would develop quite a lot of fatigue resistant, fast twitch muscle. In a chicken, the muscles of the breast are white because that's a flight muscle. Flying requires faster muscle action than standing for a chicken. I'm just using chickens because most of you have seen the white and the red muscle of a chicken, even just from the supermarket. Okay, questions about the types of muscle? We'll unpack some of these words. So, we're going to talk about myosin soon. Oh, so here's a cartoon of a muscle that is a mixture of red, slow twitch muscle, fast, white, fatigable muscle fiber, and a fast, fatigue-resistant muscle fiber. So most of our muscles are mixtures, but the proportions vary depending on the muscle. So our postural muscles are mostly slow, oxidative muscle fibers. The muscles in our eyelids are mostly fast, fatigable muscle fibers. A few red ones, but mostly white. The muscles in our legs, this is probably a leg muscle, is a mixture. So no one muscle has one fiber type. Okay, so now we're going to start to tell the story of muscle contraction. And you'll remember that muscles are innervated by motor neurons. Okay, so here is one motor neuron that is innervating a set of muscle fibers. The set of motor fibers, I want to define a term because we'll get to that in a few minutes. The set of the set of muscle fibers that is innervated by one motor neuron is called a motor unit. So one motor unit is the set of muscle fibers innervated by one motor neuron. So this is one motor unit, and here are two motor units conveniently colored to show the set of fibers innervated by one motor neuron and the set of fibers innervated by another motor neuron. Each muscle, so each muscle fiber is innervated by one motor neuron. Each muscle is innervated by many motor neurons. Okay, so now we're zooming in on the neuromuscular junction. The junction, the point of contact between the motor neuron and the muscle. So let's get oriented. Here is the nerve terminal, where neurotransmitter is going to be released. Here's the muscle cell down here. We don't see any of the myofibrils yet because this is so close to the surface. This area here is the synapse, is the synaptic cleft. This is the synaptic cleft between the neuron, the motor neuron, and the muscle. And the neuromuscular junction is a well-organized, it's an extremely well-organized synapse with active zones of vesicles loaded with neurotransmitter, getting ready to dump their neurotransmitter into the synaptic cleft, and then these folds, post-junctional folds of membrane to where the receptors are. At the neuromuscular junction, the neurotransmitter, you want to write this down, the neurotransmitter is acetylcholine. And the receptor at the neuromuscular junction are nicotinic acetylcholine receptors. And you'll recall, I hope, from the beginning of this course, that nicotinic acetylcholine receptors are ionotropic, cation permeable ion channels that would allow sodium to flow in to depolarize the muscle fiber in response to the release of acetylcholine from the presynaptic terminal. So now let's walk forward with the story of, put all the pieces into play. Let's walk forward with the story of muscle contraction. So I gave you this slide, I think. Did I give you this slide? Yeah, with a small blank to write the story. So let's get oriented. Here's a cartoon of a motor neuron. It's myelinated. So the motor neuron shows the wrapping of myelin around the axon of the motor neuron. And then here is the cartoon of the presynaptic terminal. You know what that is, that's a mitochondrion. And here are the synaptic vesicles, cartooned, but what they really should look like are bundled right at the active zone with an intense density of vesicles right where they're going to be released. So this cartoon shows them sort of dissipated around, which isn't quite accurate. Here's the synaptic cleft. Here's the muscle cell, which at this scale is going to reach to, I don't know, past Tyler to Earl of Hall in this direction and up to Route 40 in that direction, right, at this scale. So the muscle is big. Remember, it's inches long when we're talking about nanometers here, just to give you a sense of the scale. So this is something like 50 nanometers, and this muscle cell is half a meter long. So a billion times, it's a billion times longer than this distance right here, just to give you a sense of the scale. Here are the junctional folds in the cartoon. Blue is illustrating where the nicotinic acetylcholine receptors are, and green is illustrating where voltage-gated sodium channels are. Predictably, you can imagine we're getting to an action potential. So we're circling back, once again, to the beginning of this course. So let's talk. Can you raise one of these screens? Do my trick here of writing on the board. So let's tell this story. What is the story of neurotransmission at the neuromuscular junction? Okay, somebody get us started. Well, I'll get us started. The story starts with an action potential in the neuron, in the axon. Right? Action potential. That's not A and P, that's action potential. That's you. Then what happens? Okay, why? Okay, we got, there's a word that I was looking for before. You're right. The next thing is voltage-gated calcium channels open because the action potential depolarizes the presynaptic terminal, and that opens voltage-gated calcium channels. Okay? Calcium flows in. Okay, so now we've got calcium going into the presynaptic terminal in response to the action potential that came in. This is all review, but it's important. Okay, what does calcium bind to? Synaptotagmin. So now we have calcium synaptotagmin binding. Causes what? Conformational change, there's the word. Conformational change of the V snares and the T snares complex. Okay, I'm just leaving out some words because we know what they are. Okay, conformational changes of the T and V snares causes what? Vesicle fusion. Which dumps the acetylcholine into the cleft. Acetylcholine is the neurotransmitter. Okay, what's next? Okay, diffusion and binding. Diffusion and binding of acetylcholine to the nicotinic acetylcholine receptor. Nicotinic acetylcholine receptor. Okay, what's next? Okay, we got a few things that happen before depolarization. I want to unpack that.
The voltage gated
We,
channel.
there's a conformational change of the receptor, a channel opens a cation channel. So it's sodium and potassium, so sodium influx, sodium flows in, and that causes the depolarization. Okay, what's next? I'll give you a hint. We have to get an action potential. So remember, we have gone, here, this is presynaptic. Acetylcholine, this is in the synaptic cleft. Now, where are we? We're in the muscle cell, binding of acetylcholine to the nicotinic acetylcholine receptors that are in the muscle cell. So we've gone across the synapse. Conformational change of the receptor opens a cation channel, the receptor, sodium influx through the receptor, depolarization of the muscle cell. Be clear here, we're talking about, we've gone to the next cell in the synapse. And that does what? We're going to get to the T-tubules, but we don't have to get there yet. But that's a good point. But let's, what does this so this depolarization open voltage gated calcium channels? What does this depolarization open?
So this depolarization in the muscle cell
Yeah.
is happening
It's
at
happening,
like all of the receptors, right? So
it's happening across this entire synapse.
Is it still just like a small local
Yeah, we're talking about a local depolarization right here. That's right. Because we've opened these receptors, we've opened up the receptors in the blue area. So this area here has depolarized, but we need to spread it. That's what the action potential is going to do. So what does this depolarization do? Open voltage gated sodium channels. Voltage gated sodium channels. Okay. Now, this causes more sodium influx, causes more depolarization, causes more voltage-gated sodium channels to open, causes more sodium influx, right? You remember that positive feedback loop of depolarization, opening voltage-gated sodium channels, causing sodium influx, opening more voltage-gated sodium channels. There's the action potential. That's it right there. So let's just summarize that we form an action potential in the muscle, which now spreads across the surface of the muscle just exactly the way it would spread in a unmyelinated axon. The muscle cell has voltage-gated sodium channels throughout across its entire length, from South Camp, from the football field up to Route 40. Voltage-gated sodium channels all across the membrane. We've formed an action potential that propagates along the surface of the muscle, just like in unmyelinated nerve. There's no myelin on the muscle, so it's a continuous process like an unmyelinated nerve. And so this action potential muscle propagates across the entire muscle. Including the T-tubules. It goes into the T-tubules. So we'll get to that in the next picture. So you have the story now written down in your notes. But in case you missed it, the story is there in the PowerPoint that's available to you. Let's just see if we miss anything. Action potential in the neuron, depolarization, voltage-gated calcium channels, calcium influx, synaptotagmin, we got all that. Conformational change of snares, we got that. Release, fusion is release of acetylcholine into the synaptic cleft. binding, we forgot about diffusion, but there it is, diffusion and binding of the acetylcholine to the nicotinic acetylcholine receptors, conformational change, receptors open, influx of sodium, we did that, depolarization, we did that, activation of voltage-gated sodium channels, we did that, more depolarization, okay, action potential. Action potential propagates along the entire length of the muscle. So this one microscopic region, the synapse, triggers depolarization of the entire inches-long muscle cell, muscle fiber. And the action potential propagates into the T-tubule. So muscle cells have a specialization in their surface membrane called the T-tubule. These tubes that penetrate in from the surface towards the center of the muscle, where the myofibrils are for contraction. So now we got to talk about, you'll remember from cell-phys that calcium is the trigger for contraction of skeletal muscle. Just like calcium is the trigger for neurotransmitter release, calcium is the trigger for contraction, but the molecular mechanisms are going to be different. We do not have synaptotagmin in muscle cells, but we do have a calcium binding protein that responds to the change in calcium. So let's talk about calcium. This part is supposed to cartoon, so here's the surface of the muscle. Here's a cartoon of the T-tubule. It actually goes all the way, very, very deep into the muscle, not shallow like this. This is a cartoon of the sarcoplasmic reticulum of the muscle, which is where calcium is stored. And you'll remember that the SR is where SERCA is. SERCA is the sarcoplasmic endoplasmic reticulum, calcium ATPase. SERCA is what pumps calcium into the sarcoplasmic reticulum. So the calcium is high in the lumen of the SR, and it's low in the cytosol when the muscle is relaxed. Okay, so when the muscle is relaxed, low calcium, muscle's not contracting. The muscle is told to contract by the motor neuron. All those steps that we just talked about. Action potential propagates into the T-tubule, and now the depolarization of the let me do this over here. The depolarization of the T-tubule, this is the T-tubule, and this is the SR membrane. So the SR and the T-tubule are physically very close. In fact, they're connected to each other by a molecule. In the T-tubule, there is a voltage sensor, very similar to the voltage-gated calcium channel, but it's not actually acting as a voltage-gated calcium channel. It's just acting as a voltage detector. And so it detects the action potential that propagates into the T-tubule. And it undergoes a conformational change in response to the depolarization. Like the voltage-gated calcium channel undergoes a conformational change in response to a depolarization. So this voltage sensor is also called the dihydropyridine receptor. Dihydropyridine is a class of drugs. And so this voltage sensor, you can either call it a voltage sensor, which is a physiological description, or you can call it a DHP receptor, which is a pharmacological name, and they're both correct. Yeah. So here's the T-tubule, here's the SR, here's the zoom in at this contact between the T-tubule and the SR. So here is the lumen of the T-tubule, and here is the lumen of the SR. And here is this molecular space between the T-tubule and the membrane of the SR. Yeah,
So when the muscle cells had undergone that initial action potential,
Yeah.
was that at the surface of the muscle?
yes,
cell?
it is. But the T tubule is contiguous with the surface membrane. So the action potential is propagating along the surface membrane and then propagating into the T tubule and then propagating out of the T tubule and continuing along its way.
But normally an initial action potential will not be sufficient to reach all the way into the.
Oh, yes, it will. Because there's voltage gated sodium channels here.
No, I meant to the stuff that usually be activated by the calcium.
Say again?
We can't, never mind.
Okay. So, in the relaxed state, the calcium is in the SR. Now we excite the muscle, an action potential, conformational change of a voltage sensor that is physically connected to a calcium release channel in the SR. So the depolarization, conformational change of the voltage sensor, conformational change of a calcium release channel. Calcium opens the calcium release channel, and calcium escapes from the SR into the cytoplasm. Voltage sensor, calcium release channel. Voltage sensor on the T-tubule membrane, calcium release channel on the SR membrane, physically connected to each other. The names are, you can either call them voltage sensor, physiological name, or DHP receptor, pharmacological name. You can call this one calcium release channel, physiological name, or the ryanodine, another drug receptor, pharmacological name. Whatever name you want to call it is fine. They're both correct. You can use the physiological names or you can use the pharmacological names. We're not going to get into the molecular mechanisms of dihydropyridines or of ryanodine because this is a physiology class, not a pharmacology class. But the names that you'll see in the book are often the pharmacological names. That's why I'm doing this. Shad?
I have two questions. The first one, the ryanodine receptors. So you said earlier that it is the calcium release channel.
Right.
Is that just an extension of the channel that is connected to the DHP receptor?
So, yes. So there is a part of the calcium release channel, a domain of the calcium release channel that is physically in contact with domains of the voltage sensor. And so when this undergoes a conformational change here, the channel opens, calcium is released.
And can you show in this picture how the post-junctional folds are on the membrane?
Oh, they're off the scale here. So,
So if we were to
what would be
say-
the scale here? So, a post-junctional, if the synapse were right here, it would be about this big, and yeah, it'd be about this big, and the neuron would come in here and make the synapse right here. But the synapse in this picture is off the scale because the action potential has moved inches, maybe, from the synapse to this location.
Because in the previous picture, the way it's depicted, it's like as if the post-junctional folds are like almost embedded in the muscle, like
Yeah,
the
these
microscopic
are very
picture.
small. So this is a post-junctional fold,
Yeah.
which is nanometers in length. The T-tubules would go down into the basement on this scale.
Okay. So if we were to like draw
If you wanted to draw
small ones, it would be like little
tubes
insertions.
that go off the
Okay.
scale of this, but much longer. They basically, because this muscle cell is extending a mile in either direction on this scale and is extending meters in this direction on this scale, and the T-tubule, the little tubule, is going to go very, very deep. So the T-tubule is much longer than the post-junctional folds. Does that help get the scale? Yeah. Yeah. You have many, many T-tubules for each myofiber. Thousands. You have about as many T-tubules as you have sarcomeres. Thousands of them. Yeah.
I guess my question was, if the action potential did propagate to like the deepest part of the muscle,
Yeah.
could that affect anything with like the tropomyosin or anything with that complex?
The tropomyosin is not voltage dependent. Tropomyosin is calcium dependent. So there's directly the answer to your question is no. Indirectly, the answer to your question is yes. The change of the activity of the voltage sensor and the calcium release channel releases calcium that is going to trigger conformational changes in tropomyosin.
Okay, so that's why it only needs to be relatively surface level.
It needs to be what?
Relatively surface level, the action potential.
Well, remember, these myofibrils are going all, we want to be able to contract all the myofibrils, not just the surface myofibrils. So we have SR all the way deep into the muscle, and the T tubules go deep into the muscle, so that we can so that we can activate the myofibrils deep into the muscle at the same time we activate the myofibrils at the surface.
It's still on the surface of the T tubules.
That's right, surface of the T tubule, but the T tubule is going deep into the muscle fiber. Okay, so we have released calcium from the SR. Calcium in the cytoplasm has gone way up. Now, the calcium binds to something. You could have predicted that that was happening. And what the calcium binds to is troponin. You'll remember, I think, that name from Self-Is. Troponin is the calcium sensor in muscle cells, just the way synaptotagmin is the calcium sensor in neurons, in the presynaptic terminal. So calcium binds to troponin and causes a conformational change that I'll describe more in the next slide, that allows contraction. When the muscle contracts, so let me just demonstrate. I'm contracting my biceps, moving my radius and contracting the biceps. Calcium is being released into the muscle fibers of my biceps, triggering, so action potential in the motor neuron, calcium release from the SR, triggering the contraction that we'll get to in a minute. Now I am relaxing my biceps. My biceps are being stretched. My triceps are activated, right? So when I do this, I am contracting my triceps and stretching my biceps. And that requires the calcium not to be in the biceps muscle. In order for the cell to be relaxed and stretched, the calcium has to get taken out of the cytoplasm, and that's the role of CERCLA, to pump it back into the SR so that the muscle can relax. Remember that muscle contraction is what we're focused on, but for every muscle contraction, there's a muscle relaxation, or otherwise we're in rigor mortis. Okay, so we're going to talk about the molecular mechanisms of muscle contraction. Let's take a break, and we'll come to this, because this now is going to be very reviewy from Self-Is. Yeah. Into contraction of the myofibrils. This is review from Self-Is, but I'll go through it. Calcium binds to troponin on the thin filament. Calcium binds to troponin on the thin filament and is going to allow the thick filament to motor along the thin filament and make the contraction. So let's unpack that. The thin filament of muscle is actin, troponin, and tropomyosin, organized into a filament. Actin, troponin, and tropomyosin in a filament. Can't come up with a synonym for filament. A molecular line. The thick filaments are made up of bundles of myosin. The myosin heads are facing out from either end of a bundle. So here's the myosin molecule with heads and tails. The tails bundle together in a coiled coil connection, and they have a middle, and the heads are pointing out in both directions from the middle in this myosin bundle called the thick filament. So here is a electron micrograph of a muscle, of thick and thin filaments. So the thick lines here are thick filaments. The thin lines between the thick lines are the thin filaments. And the cross hatchings are the myosin heads connecting the thick filament to a thin filament. Those connections, those heads, are called in many textbooks, most, I think your textbook also, called cross bridges. So the myosin head makes a cross bridge between the thick filament, which is myosin, and the thin filament, which is actin, troponin, and tropomyosin. In low calcium, so this cartoon is not great, but the words I think are helpful. In low calcium, troponin is in a conformation that prevents the myosin head, from interacting with actin. In low calcium. So in a relaxed state, the myosin head can't interact with actin. And so this fiber, this filament, is not connected to the thick, the thin filament is not connected to the thick filament, and the muscle can be stretched because the filaments are not connected, because troponin is in a conformation that prevents the heads from interacting with the thin filament. When calcium is released from the SR, going back to the story, I'm not going to say it again, but calcium is released from the SR, calcium binds to troponin, conformational change of the troponin moves the troponin and the tropomyosin out of the way, conformational change of the troponin and tropomyosin, exposing the sites on actin to which myosin can bind. Now myosin can bind to actin, and so it does. And so now the thick filament and the thin filament are connected by thousands, maybe millions, of heads, of myosin heads. And now this muscle is getting ready to contract. And I'm going to get into the molecular mechanisms of contraction next. So let's walk through this. Here is a thick filament. Here is a thin filament. The thin filament is connected to the end of the sarcomere called the Z line, or Z disc. Okay? So the thin filament is connected to a big part of the cell. So and I'll unpack that in the next slide. Here's the thick filament with just one of the millions of head groups that it has. It's got millions of them, but we're just looking at one. In low calcium, so this is a relaxed resting muscle, low calcium, the myosin head cannot interact with the actin. So this filament, the thin filament and the thick filament, can slide past each other because they're not connected. Sliding filaments. So when I stretch my biceps, the thick filaments and the thin filaments are sliding past each other. because they're not contracting. When I contract my biceps, the thick filaments and the thin filaments, the thick filaments are pulling on the thin filaments to cause the contraction. So sliding in relaxed state, motoring in the contracted state. Sliding in the biceps, motoring in the biceps. So now let's talk about the motor. Calcium calcium moves troponin and tropomyosin out of the way. Myosin interacts with actin. When myosin interacts with actin, the ADP and phosphate that were part of it dissociate, right? So this binding causes a conformational change, lowers the affinity for ATP and phosphate, all part of the conformational change. ADP and phosphate dissociate because the affinity has just decreased. The dissociation of the ADP and phosphate that used to be attached to it, and we'll come around to the cycle, causes the head to go from a extended configuration to a bent configuration. So the head is attached to actin. Now the dissociation of ADP and phosphate causes the head to go into a bent configuration, and you can see it's going to pull on the actin. That's the myosin motoring right there. It just pulled this part of the muscle in this direction. Conformational change. This conformation of the myosin has a high affinity for ATP. So as long as there's mitochondria or glycolysis making ATP, ATP binds. ATP binds. ATP bound flexed myosin has a low affinity for actin, so it lets go. Now, you'd say, well, then now the muscle is stretching? No, we're still contracting. It's letting go for a microsecond. Then it's going to hydrolyze the ATP to ADP and phosphate. There's where the ADP and phosphate that it let go of came from. The ADP and phosphate now, it's in a configuration that causes it to go from the bent configuration to the extended configuration. And now it's going to bind to a new spot on the actin, because previously it pulled the actin, and now it's going to bind to a new spot on the actin and do it again. So doing that over and over and over again pulls the myosin, thick filaments, pull the thin filament towards the middle of the sarcomere. So we do this cycle a few thousand times, and the muscle gets shortened. Now we stop. The nervous system says, okay, don't do that anymore. No more calcium released from the SR. Calcium gets pumped into the SR. Low calcium. Myosin lets go. Myosin lets go of actin and can no longer bind because the troponin is now in the way. And now we can slide those filaments back to their length, their long configuration. Okay, so this is, I think a lot of this should be reviewed from self-is. Yes, Leah?
Just to be clear, like, so this, when we're contracting the muscle, this is the same that's happening so many times, like even to just contract.
So when I do this, I am running this cycle a few thousand times.
Okay, cool. But then when it's relaxed and stuff, it's like the filaments are not interacting.
That the filaments of my biceps are not interacting anymore. My triceps are doing the contracting, and they are causing the filaments of my biceps to stretch past each other and to reach to slide past each other into the stretched, elongated length. So this is the molecular mechanism of contraction. Now, this slide has an interesting side note here. Let's go through this. So the muscle dies. dead muscle. Right? The person is no longer breathing. No more oxygen. No more oxidative phosphorylation anywhere. Low ATP, right? So in a living muscle, we need high ATP to do this, because every time it does that, to contract this muscle, it's running the myosin, this cross-bridge cycle 1,000 times, and for each of those, it needed to bind an ATP and hydrolyze it, and then release the ADP and phosphate. So an ATP molecule is required for every run of the cycle. In the absence of ATP, like absence of oxidative phosphorylation, two things are important. One is CIRCA is a calcium ATPase. Pumping calcium into the SR required ATP. So now calcium stays high in the cytoplasm. And so this machinery is now uncontrolled because calcium is just being leaked from the SR into the cytoplasm and can't get re-pumped up, can't get re-taken up because there's no ATP to run the pump. So the muscle contracts. And the ATP, remember ATP was required for the myosin to let go of the actin in the middle of the cycle. But without ATP, the myosin cannot let go of the actin. So it stays attached. And now we have the rigor mortis. So muscles, so when an animal dies, the muscles contract sort of uncontrolled, and then they reach a state of permanent contraction for a couple of days or weeks, and that's called rigor mortis, rigid death. And so let's see, how many of you ever seen a fairly intact dead deer on the side of the road? What is the position of their head almost always, if it hasn't been completely messed up by the car that killed it? The deer's head is almost always bent back like this. And in fact, when you see fossil animals, like fossil dinosaurs, their heads are bent back like this because the muscles of their neck have undergone contraction during death. So they went through contraction, and then they went into rigor mortis. We don't do that so much because we have muscles in the front of our neck that are almost as strong as the muscles in the back of the neck. But for quadruped, that has to hold its head up, even though it's horizontal, it's got strong muscles on the back of its neck. And so those muscles, when they contract, they tip the head all the way back. They extend the neck completely. So rigor mortis, in the absence of ATP, is a high calcium, low ATP cytoplasmic environment that causes contraction and then attachment of all of those myosin heads so that it can't be stretched anymore. And that's, the body in the cadaver lab was in rigor mortis and then was fixed in the embalming, and so it's sort of in a permanent rigor mortis because the embalming process fixed the muscle in the rigid state. That's why we can't move the elbow of the cadaver very well, because the muscle can't be stretched. We can stretch our muscle. We can't stretch his muscle because it went into rigor mortis and then it was fixed by the embalming process. So here, let's put this together and use some of the anatomical terms of a muscle. So this anatomy is testable. So here is a so this is an electron micrograph of a muscle cell, muscle fiber, one sarcomere of a muscle fiber. Thousands of these stacked end to end to make, and actually scores of them stacked side by side to make one muscle fiber. Thousands of them this way, scores of them this way to make up one muscle cell. The membrane, in this case, is, off the top. We're inside the cytoplasm of this cell. One sarcomere. One sarcomere is defined as going from the Z line to the Z line. So the Z lines, or Z discs, interchangeable name, the Z lines or the Z discs define the end of a sarcomere. The thin filaments are connected to the Z lines. So here's a thin filament, thin filament, thin filament, thin filament connected to the Z line. There are proteins that do this connection that we're not going to name. The thick filaments are interdigitated, thick filaments, thin filaments, interdigitated with the thin filaments. Thick filaments, so thin, thick, thin, thick, thin, thick. The middle of the thick filament is called the M line. The band on either side of, the band on either side of the M line that has just thick filament and no thin filament is called the H zone. Okay, so this we didn't teach in cell phys because this is anatomy, cellular anatomy of a muscle cell. So now we're moving past cell phys. M line, the naked thick filament on either side of the M line is the H zone. Now, on either side of the H zone, where the thick filaments are in contact, could be in contact with thin filament, is called the A band. So A band is on either side of the H zone. And that's where there's thick and thin filaments. That's where the motoring takes place, where there is contact between myosin heads, and you can see the thousands of them here, and the actin filaments. Where there is, where the Z line has only thin filaments, where the thick filaments do not reach all the way to the Z line, is called the I band. So on either side of the Z line is the I band. So walking from one end of the, so that I've been starting in the middle and going to the out. Now let's just walk from one end to the other. Z, I, A, H, M, and then it's palindromic. We would do the other thing on the other side. Many years ago, a student came up with a mnemonic, which I thought was actually pretty good. So Z, I, H, no, Z, I, A, H, M. She came up with Zeus is a heroic man as the mnemonic. Now, of course, it's false. Zeus isn't a man at all, but, you know, whatever. Yes?
This is probably fine, but I just want to make sure. The I band to me looks like it comes before the Z line, like it's connecting the two, like, sarcomeres now, like a new sarcomere, like, so why would you?
That's right.
No.
So the I band is on either side. So this is the I band of this sarcomere, and this is the I band of this sarcomere.
Okay, gotcha.
This is a different sarcomere. This is the I band of this sarcomere on this side, and here's the I band of this sarcomere on this other side. So now you can, question? Does the A band include the H zone? Technically, yes.
How much of the H does it include? Half?
How much what?
How much of the A band is? You have two A bands there, each one.
The A band is the length of one thick filament, or is the length of the thick filaments. So now you can see, so let's now put this together into contraction. Here is a relaxed muscle that can be stretched. Right? Z, I, A, H, M, just in this half of this sarcomere. If we pull on this, we can pull the Z discs apart from each other and stretch the muscle. Because it's in low calcium, the myosin heads have ADP bound, but the calcium is preventing them from interacting with the thin filaments. So the thick filaments and the thin filaments are not interacting because calcium is not present enough to move troponin and tropomyosin out of the way. So these filaments can slide past each other. I can stretch my biceps. When this sarcomere gets the message to contract, calcium goes up, calcium moves troponin out of the way, the myosin heads on this side do their, well, let's go through it. So binding, so imagine I'm a myosin molecule. You have to imagine there's a thin filament here. Calcium has now removed the inhibition. Binding, well, sorry, I need to be in the extended position. Binding, ADP and phosphate release, power stroke. Binding of ATP, unbinding of the myosin from actin, ATP hydrolysis, binding, right? So it's doing the cross bridge, and I'm doing this on either side. Right? I'm binding, and so I'm pulling the Z discs towards each other. So what contracts are the I band. The I band gets shorter, and the H zone gets shorter. The A band, which is defined by the length of the thick filament, does not get shorter. The Z disc is not that's just a disc. It's not a length. So the myosin heads are pulling the Z discs towards the M line. And the muscle gets shorter and contracts. That's muscle contraction. So I'm going to leave this as a homework problem. So here are the steps, and you can do this at home, putting them in the correct order. The answer is on Moodle. So let's end with one more key concept. We're almost done for today. You know that you can control the strength of your muscle movement. You know that lifting a pen requires less force than lifting lifting my hand requires less force than lifting the computer, right? I should have brought a weight in here. I should have brought a lead weight or something. Lifting my hand takes less force than lifting the cup of coffee. So you can control the strength of contraction, even though that's not the same. There are two ways that we control the strength of contraction. One is temporal summation, the frequency of action potential. So one action potential creates one pulse of calcium, creates one twitch of the muscle. Two action potentials, two pulses of calcium, two twitches that are summed. They're temporally summed, they're on top of each other in time. 4 action potentials, 4 pulses of cytoplasmic calcium, 4 twitches summing to each other. Now 8. So if we, 1 action potential, 1 twitch, a string of action potentials, we get to a steady state. The higher, so 1 action, or 1 twitch, summation, summation, summation. The higher the frequency of the action potentials, determined by the motor neuron, which is connected to your brain eventually. Low frequency, low force. Moderate frequency, moderate force. High frequency, high force. Temporal time summation. So the higher the frequency of action potentials in the motor neuron, the stronger the contraction due to temporal summation. In addition, this is the last point of the day, there are spatial summations. Your brain is figuring out how much force do I need to lift this object. And it's saying, okay, I'm going to need to activate so many motor units. I know that lifting a pen only requires a few of the motor units of my biceps to work in order to lift it without throwing the pen across the room, right? If I do this as hard as I can, I'm going to throw it. I'm not going to do it because it'll break. But I also know that if I want to lift a heavy object, so heavy I can't lift it. If I want to lift a heavy object, so now I'm going to try to lift this, and my brain is saying, you're going to need to work. Not only are you going to have to maximize the frequency of all your motor units, but you're going to have to engage more motor units. Not just one, but maybe all of the motor units you've got in your biceps in order to lift this chair. My brain has told me, you need to work hard, and so I do this, and so I knew, and I was getting feedback. When I started this, I was saying, okay, I need more, I need more, I need more, I need more. Okay, there's the number of motor units I need to lift this object. Okay, so your motor system is telling your muscles how many motor units are necessary to contract. You've probably all had the experience of, like, imagine there's a styrofoam brick, and you lift it up, you think it's a brick, so you're engaging a bunch of motor units because you think, okay, this is going to weigh something, and then, oh, it didn't weigh anything. Your brain was tricked by the fact that the brick was styrofoam and not cement. That was your brain being tricked into engaging more motor units than you needed for that contraction. So temporal and spatial summation are what's required for grading the strength of muscle contraction. And that's where we'll end today, and then on Thursday we'll come back and start talking about the higher control levels of the motor system.